Hello everyone, welcome to another MCAT ACE lecture. So here we are doing the second part of the metabolic pathways that we've been describing for glycolysis, fermentation, citric acid cycle, and electron transport chain. These were all covered in the first lecture. Now in this follow-up lecture, which is the second part, we are now going to cover the pentose phosphate pathway, gluconeogenesis and glycogenesis, regulation of these pathway, and then finally conclude the lecture with metabolism of fatty acid and proteins and some practice questions in the end. So this is a high yield drawing that I've shown you at the beginning of the first lecture. So basically glucose is rich in potential energy, and we've discussed how through glycolysis and through aerobic respiration it can yield 36 to 38 ATPs. And then depending on the source that you're reading, whether it's NADH that is giving 2.5 ATP versus 3 ATPs, it could also be 30 to 32 ATPs. So the top one is assuming NADH gives 3 ATPs and every FADH gives 2 ATP. The lower one is assuming 2.5 ATP per NADH molecule and 1.5 per FADH molecule. That's going through aerobic respiration. Under anaerobic reduction, however, there would be only 2 ATP that is produced throughout the glycolysis. So we've already covered this topic extensively, describing how many ATPs, which stands for adenosine triphosphate, will be produced through each one of these reactions and the contributing factors that can affect the rate of all these reactions. Now, next we would like to discuss the role of glucose as a precursor for supplying different metabolic intermediates for biosynthetic pathway. So we will discuss how glucose can be used as a storage, can be in the storage form like glycogen, starch, or sucrose, or it can be used in the other pathways like pentose phosphate pathway for production of ribose 5-phosphate, which is required for DNA, RNA, and nucleotide synthesis. And this pathway also produces NADPH, which is required for synthesis of steroids, fatty acids, and the reduced form of glutathione, which is required for defense against oxidative stress. Now here I'd like to point out not to confuse NADPH with NADH. So if you recall glycolysis and then citric acid cycle, they were all giving us 10 molecules of NADH per 1 glucose molecule that was going throughout this reaction. Here we are talking regarding the pentose phosphate pathway, we are talking about NADPH. So this molecule has an extra phosphate and it's different than NADH. NADH is used for energy production or ATP / adenosine triphosphate production, while NADPH is used for different biosynthetic pathways. So that's one point I'd like you to pay attention to. And if you like to remember that pentose phosphate pathway is associated with NADPH, you can always think of pentose phosphate pathway is associated with production of NADPH. All right, now with that background information, let's move on to the actual pathway. So here we have the pentose phosphate pathway, the other name for which is hexose monophosphate pathway or shunt. So hexose mono pathway. So sometimes you hear the HMP pathway. That's what we are referring to, pentose phosphate pathway. And so here you can see that in this reaction, glucose throughout the glycolysis will be converted to glucose 6-phosphate. That's the first reaction, and that was via the hexokinase. These were all reviewed in the first lecture. Now, throughout the first reaction of the pentose phosphate pathway, the enzyme glucose 6-phosphate dehydrogenase will convert glucose 6-phosphate into different intermediates, as a consequence of which there will be production of 2 NADPH and 1 ribulose 5-phosphate molecule. And then the ribulose 5-phosphate will be later converted to ribose 5-phosphate, which is required for production of nucleotides, RNA, and DNA. while the NADPH is used for production of the reduced form of the glutathione, steroids, as well as fatty acids. Now, one more point about the glucose 6-phosphate dehydrogenase. Not only this is the first reaction of the pentose phosphate pathway, this is also the rate limiting step. And so if NADP plus concentration is too high, then it would have a positive effect on the glucose 6-phosphate dehydrogenase in order to produce more NADPH. And then if there is too much NADPH, then there would be a negative effect in order to lower the production of the NADPH. Now, if you've noticed, the reduced form of glutathione is being produced via the G6PD reaction. So glucose 6-phosphate dehydrogenase is required for production of GSH, which is the reduced form of glutathione that is helpful in defense against reactive oxygen species. So what happens in patients who don't have this enzyme? Therefore, they would be susceptible to attack by reactive oxygen species, and so these patients can develop hemolytic anemia from any compounds that can cause reactive oxygen species, like for instance sulfa drugs can cause hemolytic anemia in these patients. Alright, now moving on with the pentose phosphate pathway, there is actually two different phases. One is the oxidative phase, which I just described for you, and the net reaction is glucose 6-phosphate plus 2NAD plus H2O will yield ribose 5-phosphate, a CO2 molecule, and 2 NADPH plus 2H plus. So this is basically the net reaction.